Hello, Genetics 311 students. Today we are going to be talking about prokaryotic DNA replication. So our first step is going to be initiation. Here is our prokaryotic circular DNA, and this is our origin of replication. This initiator protein is going to come in and bind to the replicon, causing unwinding and a short stretch of single-stranded DNA, and also our replication fork right here. Our next step in prokaryotic replication is unwinding. So this is when helicase, which is given right here, attaches at the replication fork and moves into the fork, breaking the hydrogen bonds, and then we have our single-stranded binding proteins, which will coat the single-stranded DNA to protect it and prevent double-stranded DNA, also known as hairpins. And then gyrase over here is a topoisomerase that relieves the supercoiling, which we will see in the next step. So for our last step, which is elongation, RNA primase is going to come in and lay down RNA primers on our lagging strand and our leading strand, but mainly on our lagging strand because it moves from 3' to 5' end, which is going to be discontinuous replication. And then DNA polymerase 3 will come in and add nucleotides to elongate the DNA from 5' to 3' end. It also, when it's adding the nucleotides, creates phosphodiester bonds between the two nucleotides. And then our next player is DNA polymerase 1, which aids in removing the RNA primers with 5' to 3' exonuclease activity. However, if this weren't to perform the way it's supposed to, it would cause a lot of wrong nucleotides within, and the wrong nucleotides will not be replaced by the right ones, and it could have an impact on the RNA, causing mutations that can be very harmful. But if it were to perform correctly, which in this example we will be saying it does, DNA ligase will come in and seal these nicks that are created from the Okazaki fragments.